All right. So thanks everyone. That's our second and last talk of the day. Karim was very kind to give us a little introduction to magnetoencephalography. Lots of people in the room don't know anything about neuroscience or neuroimaging.
Most people.
I think.
I don't know much about neuroscience myself.
There are some engineers in that room.
I can attest to that. Yeah, I know nothing about the brain.
Me neither. So we're all the same level.
Anyway, so hopefully we're gonna get a great little introduction and maybe some pointers to a few tools.
Yeah.
I don't know, you get the pointers?
Yeah.
Wonderful. So Karim.
Mostly logos. People will find the logos on the internet.
And the logos.
Yeah.
Thanks so much for accepting to come. And
thank you so much. It's a pleasure to be back to BrainHack School. So I think I'm going to discover the content of this talk as we go along with you, because I've made most of it in the last couple of hours. So I threw. Can you hear me? Yeah.
Yeah. The microphone, camera, because we're recording. So
Hi,
if you
Mama.
move too much, it's going to be very like
Okay.
people are going to feel sick watching you.
Okay. I'm not going to move.
Sorry about that.
Oh, good. So the idea was to give you just some, let's say, superficial information related to the electrophysiological side of data processing when it comes to the brain. I'm sure you've heard already. I think you had an introduction today from Basile about processing fMRI data, and so the idea now is to give you just some insights into the type of signals we deal with when we record the electrophysiological components that come from the brains. So basically the way I'm going to cut this is in two parts. In the first part, I'll give you some information about EEG, MEG, so electroencephalography and magnetoencephalography, specifically tailored to those of you who have never seen this before. Those of you who have seen this will probably get bored of that. And the second part is going to be just a presentation of some of the tools that I suggest you look into, specifically during your projects here, if you plan to work with EEG or MEG or intracranial EEG data. I'd like to start off first with a quiz. There'll be one quiz and two announcements. So this is the first quiz. These are techniques, and my question is, what do these three techniques have in common? So somebody wants to take a guess? Don't tell me the three of them are smiling.
They look really comfortable.
They look
Really comfortable.
comfortable. Yeah, it could be one explanation. Somebody else has an idea?
Joke. Does it count? Can I say it?
You can.
These are all tools for errors, right?
Yes. So this is actually so there's nothing to do. The common thing is that none of these has to do anything to do with brain signals. Actually, these were 1934, 905, and 23, and this is from Carl. I think his name is Carl Ludwig Nessler, also known as Charles Nessler, and he developed the first perm machine to actually give you curly hair. So this was the very important information of the day. Now you know this. Now you're much more knowledgeable than you were about 30 seconds ago. Okay, now that you're even more knowledgeable, I'm going to tell you about brain imaging technology. And most of you know that we have a large array of tools at our disposition today that we can play around with when we want to look at what's happening in the brain. You might be familiar with most of these. So you see down there, you see this is a CT scan, infrared spectroscopy, an MRI machine, here an MEG machine, we'll talk about that more in a while, and an EEG system. Also positron emission tomography, PET machine. So we have many tools that we can use when we want to tap into what's going on in our brains, both for healthy individuals and also in patients. And these tools differ in many ways. Some of them are well suited to look at anatomy of the brain, others for function. Some of them are invasive and others are non-invasive. Some of them have really good temporal resolutions, and others have a really good spatial precision. And why is it so? Why do you have all these differences? It's because mainly these techniques differ in the underlying brain signal that they detect. Okay, so they're not looking at the same aspect of brain activity. On one hand, you have the brain's electrical activity, and on the other hand, you have brain's vasculature or blood flow. So if you're looking at electrophysiological responses, this is basically what we'll be recording with EEG, MEG, and intracranial EEG. And this is a direct measure of neurons activity. Neurons have this electrical activity. Action potentials are fired in the brain. This gives rise to an electrical voltage difference and also to current flow. So this is nicely detected with EEG, MEG, and intracranial EEG. And on the other hand, you have the ability to detect hemodynamic or metabolic responses. That's what fMRI and PET or NIRS are capturing, and that's an indirect measure of neural activity. I'm not saying at this point that it's worse, it's just another type of measurement that records from brain activity. One is a direct measurement of the electrical activity, the other one is just an indirect measurement because it's looking at changes in blood flow. So now let's focus on the electrophysiological side of things. You have EEG, MEG, magnetoencephalography, what we call ECOG, that's electrocorticography, and SEEG, which is stereo electroencephalography. These two together, ECOG and SEEG, are often referred to as IEEG, nothing to do with an iPhone. This is intracranial or intracerebral EEG, so they're invasive. So this is why we put them together, and also microelectrodes recording in humans are now available. So you have basically two groups, either non-invasive recordings or invasive measurements. So now, because some of you might not be very familiar with the EEG and MEG, I'm just going to give you a very brief overview of history. This started in 1924. This guy called Hans Berger invented the first non-invasive EEG recording, and he was one of the first to report oscillations in brain rhythms. So among other things, fluctuations in the 10 hertz range, that is 10 oscillations per second, and he saw that that changed when people opened or closed their eyes. So you can see here, this is like the time manuscript that he had. This was, let's see, yeah, his findings were found in 24, and he published his paper in 1929. That's 5 years later. So if this is any consolation to PhD students who see time going past and their paper's not coming out yet, it has been like this since the very beginning. It takes time. So, as I said, you see changes in eyes open, eyes closed. You see changes across the different stages of sleep and in wakefulness. And cognitive tasks also changes brain rhythms that are measured with the EEG. And so here one question one might have is, was Hans Berger really the first to record the brain's electrical activity? So this is the question I asked you. Do you think he was really the first one to record it?
No.
Some of you have this hesitation. Others know the trick. Of course, he was not the first. Why not? Because before him in 1875, there were recordings of brain activity, electrical activity from the brain, only they were not in humans and they were invasive. So invasive recordings in rabbits and monkeys before, and invasive recordings in dogs in 1890 came before. And then after Hans Berger in 1934, Edgar Adrian measured what now we refer to as alpha rhythm, which used to be called the Berger rhythm before that, because it was discovered by Hans Berger. Gray Walter in 1936 had the first EEG setup where you had more than one channel. So they had, I think he called it a toposcope, basically doing topography, looking at electrical activity across different channels. I'm going to speed up a bit. So EEG today looks quite different. This is what you see here. So we have systems that can go up to 256 electrodes. Some we have either wireless or connected setups. You can have dry or wet electrodes. And just to plug in, this is the first announcement. For those of you who might be considering at some point running your own EEG experiments, we are setting up now the ability to do high-density EEG at the UNF here. It's a 128 EGI system, and it's in the process of being set up. Okay, what about MEG? What is MEG? So the first reflex you have nowadays, if you're looking for information, of course you Google it. If you Google MEG, this is what you get. MEG Montreal Festival. It starts, I think, it starts on the 30th of August, so that's happening soon. So you still have time to go and enjoy it. It's a contemporary electronic music festival called MEG. But of course, that's not what we're looking for. So if you look harder on Google, you will find the solution, which is this. This is MEG, a science fiction movie. Also a lot of fun, but don't worry too much. Keep calm. Love MEG. Why? I'm going to tell you the very short story of MEG. It's not like EEG. EEG started a long time ago. MEG has a very, very short history. It can be summarized in these 3 dates. In '68, David Cohen made the first, at the time, very noisy recording of the brain's magnetic field. in 1970, James Zimmerman invented the SQUID. The SQUID is a superconducting quantum interference device, basically able to measure minute, very small magnetic fields. And in 72, again, the same David Cohen from 68 published his second science paper, now this time saying, okay, I managed to measure it again, but now with the SQUID. So that gave him his, the science paper on this, and this is David Cohen here, and this, the article in 1972. What does a MEG look like today? Well, this is more what it looks like today. You have systems with up to over 300 channels. And this brings me to my second announcement, of course, if those of you are interested in the future of acquiring MEG data. We also have a MEG system at the University of Montreal, a 275 CTF channel. Okay, now, a very brief introduction to the signal, the origin of the signal. It differs from what we see in other techniques, so I'm going to take a few minutes to go over this. Without going into details, the basic structure of a neuron, as you can see here, you have a cell body, you have dendrites, and you have an axon. And electrical impulses travel through the axon from one cell to the next, and then they are transferred in this way. You have a synapse, and so there's a release of neurotransmitter that happens at the synapse, and then on the other side, on the postsynaptic neuron, you will have a second potential. So you have two types of potential, an action potential that goes down here, and on the postsynaptic side, we have a postsynaptic potential. And again, one of them is very short-lived. It's transient, it's about 2 milliseconds in length. The second one, the postsynaptic potential, lasts a bit longer. This one is biphasic. This one, so we're talking again on, this is on the receiving end, so this is on the postsynaptic neuron, and this is traveling down to the postsynaptic neuron. When we record activity in MEG EEG, what you need to keep in mind is you need to have synchronous activity from all these cells, from a large number of cells that happen at the same time. So, in your opinion, if you had many of these, or many of these, which one is more likely to end up summated into a signal that is strong enough that you can actually even measure it from the surface of the brain? Exactly, it's going to be this one. Why? Because it's longer, so it's more ideal for summation. It's monophasic, so there's no cancellation, and it lasts longer in time. So when we say point, it's not actually not just one point in time, and even a one point in time probably doesn't even exist. It's a very, very short window in time. So in that very short window, you have the synchronous activity of all these cells, and for their activity to sum up, this is probably what allows the summation, because you have more chances of having overlapping PSPs. So what we think is being recorded is synchronous PSPs and a bunch of neurons, a large number of neurons, and this gives rise to signals that you can measure at the surface here with an EEG setup, for instance, and this gives you this type of traces that you see down here. So now a very quick word about pre-processing and dealing with artifacts. There are several sources of artifact. One of them is eye movement, and this can be typo here, sorry, so eye blinks, saccades. The heartbeat is also a source of artifact. Muscle activity, so you have the neck activity, jaws, and any type of movement actually that happens can lead to artifacts and signal. And then you have problems that are easier to solve, like line noise. That's a very has very clear properties. For example, it's going to be 50 hertz in Europe, it's going to be 60 hertz here, and that's easy to deal with because you can just have a notch filter and get rid of it. These other ones up here are more tricky to deal with, but we can still solve that problem. So our options are either you reject, so basically identify and then exclude data segments with artifacts that you're not able to correct. And this is generally most of the time my preferred choice, because then you are sure that your data is clean enough, but actually because you exclude any biases that could come from a correction that was not well done, conducted. But of course, to be able to have this luxury, you need to have a lot of data. If you have a lot of data, then you can probably get away with just removing segments that that contained artifacts. You can correct, so in other words, you can run ICA or PCA and then remove segments that are clearly related to, for example, eye blinks or heartbeat, and then you can exclude those wrong segments by, after the decomposition, by excluding those components. The one where we pretend to be an ostrich over here is basically putting your head down in the sand and pretending these artifacts don't exist. This is not something that I would recommend, but this is actually something that does happen. And this is something that if you have a huge amount of data, I would actually personally consider. Because I think there are ways of getting canceling out a lot of artifacts that are not linked to your stimulus. So it depends on the type of artifact that you're trying to reject. For many artifacts, rejection is easy. So, for example, heartbeat, that is something that we quite know how to deal. We know well how to deal with. Also, eye movement artifacts using EOGs are quite reasonable to deal with. But sometimes you still have some dirty signals. You still have some variable activity in there that you know is actually not neurophysiological. Sometimes, if you have enough trials, if you have enough individuals, you'll be able to get rid of it. It's going to give you an impression of what this looks like. Here you see artifacts that come from cicads. And here you have the muscle activity, EMG. This here comes from the cardiac, so this is the heartbeat. Here you have slow drifts that could come from changes in skin potential. Here you have oscillations, so this is obviously not an artifact that you're looking for. This could be something of interest. But you see there are many different signatures that differ across your data, and they're not always this easy to see in your signal. Sometimes they're deeply buried into your signal, so you need to run these techniques such as ICA, for instance. If we look at the topographies, here you see a topography for an eye blink. This is an EEG. The topography for an eye blink with MEG, this is an axial gradiometer, MEG, like we have here. And this is planar gradiometer, which you have down here. And you see that there are different topographies. So when you're working with this type of data over a sufficient period of time, you get used to even actually visually identifying in your data sources of artifact. Okay, a very quick word about data analysis. I'm not going to go into details because if I wanted to do this properly, it would take me a couple of hours just to focus on this one slide. But just so that you know, for those of you who have not seen this before, I'm sure many of you are familiar already. But basically, you have a repetition of trials. Let's assume this could be EEG or MEG data, right? One of the easiest things in data analysis for EEG and MEG is just simply take your data and average it across all your repetition of your trials. So if you have 100 repetitions of the task, you will just average your data 100, the 100 epochs over time, and this will give you what we call an evoked potential. The idea here is that anything that is phase locked to the onset of the stimulus will be preserved, and anything that comes with a jitter over time is going to be cancelled out. So anything that is not directly phase locked to the onset of your stimulus or to a button press, for instance, it will be simply cancelled out. So this gives you a nice clean ERP, and this has been shown to be very useful both for studying cognition, but also we're looking into pathologies of the brain. This has been done for many years. Over the last 15 to 20 years, there has been an increasing interest in looking at brain rhythms, brain oscillations again. And these brain rhythms, they're not necessarily always, they can be evoked by the stimulus, but they're often induced by the stimulus. So this brings us to a very important concept, the difference between induced and evoked oscillations. There's up here, evoked and induced oscillations. The way we do this is, if now, for example, let's assume I've averaged all my trials over time. This gives me my evoked potential. Now, if I take a time frequency map, a time frequency decomposition, for example, using wavelets or using the Hilbert transform of my signal, this is going to give me a map here. Across X, I have the time, and here on the Y-axis, it's frequencies. And what we see here is the power of the signal in these different across these different frequencies over time. And so you see you have an evoked blob here that reflects what's going on in this signal over here, and nothing elsewhere. Now, if we take a different path and we say, okay, now for each trial, what I'm going to do is, I'm not going to average my trials. No, I'm going to take each trial, compute a time frequency map. This is going to give you time frequency representation here. Same thing for the second one, and so on for all of these. And then I'm going to average these maps. And if I do that, I'm going to get an overall time frequency map that is average, okay, across all these frequency maps, but that differs, that does not look the same as the one I have here. Why is that? Why don't they look the same? The reason is that these are time frequency maps, so they show us the power across signal across time and frequency. So I don't have negative and positive values. I'm looking really at the oscillatory components across different frequencies, so their amplitude, these are coefficients that have positive values. So there's no cancelling out happening like when I'm averaging these time signals. So even if I have things moving around a bit, as you see this component here, here, and here, it's probably the same brain response to the stimulus. Only it comes with a small jitter. Okay? So in other words, it's not phase locked to the onset of the stimulus or to a button press or to some event, but it's always there. But it has a slight jitter. That was lost in this time domain analysis, but with the time frequency domain analysis, it's preserved. Okay? So this is one of the interesting options that we have, is to switch to the time frequency domain and look at the induced oscillations. And we do that trial by trial, and then we average our trials. Another important aspect of MEG and EEG data, and this is even more relevant for MEG, is the fact that we can go and look for use source reconstruction techniques to look at the sources that underlie the signals that were recorded. This gives us added anatomical information and tells us where are things happening in the brain, as opposed to saying it's on electrode CZ or C4 or C3. We can actually really go and identify brain areas. The only couple of pieces of information I'll give you about this is that when you're doing source reconstruction, we have two concepts, with the forward model and the inverse problem. As some of you might know, the forward model is how do I get from these activated sources to distribution on the scalp. And the inverse problem is, if I have my data recorded on the scalp, how can I go from there and come back and identify whereabouts in the brain was there any activity? Okay, and so for the forward problem, again, I won't have time to go into details. I'll just tell you that there are several modeling approaches that we can use. Basically, we model the geometry and we model the activity. We model the geometry of the brain by assuming, for instance, either a spherical head model, concentric spheres, as you can see here, or we can use realistic models based on, for example, BEM methods, boundary element methods, or we can use finite element methods and so on. So these are basically different types of discretization of a problem and turning into a modeling problem, where we can from here then, using elemental dipoles on these nodes, create based on very simple laws of Biot-Savart that tell us basically what the magnetic field looks like at different points in space if I have a source somewhere in space. So basically we're using these elemental dipoles and the geometry of the head. We can create, knowing the position of the sensors, what magnetic field I should expect. Okay? So that's the forward model, going from the sources out to the sensors. What we're interested in doing most of the time is something different. We want to go back from the sensors, where I've recorded the data, down to the sources. And so there are different families and different approaches to do this. The dipole model, or the dipole localization problem, is basically trying to find, if I have this type of data, where are the best dipoles that basically give the best explanation of the data, they explain the variance in the data. A distributed source model, on the other hand, is something where I'm going to put dipoles across all the brain. So every node on my tessellation of the brain is going to have an elemental dipole, and my problem is estimating the amplitude of those dipoles. I have the information about the location of the dipoles, and I can also include the information about the orientation of those dipoles. And then the idea is to, again, solve this inverse problem by estimating the amplitude of these sources. That's a non-trivial task. It's mathematically an ill-posed question, and it's underdetermined because of the number of potential sources that I have in the brain and the limited number of sensors that I have on the sensor. So we normally solve that by using regularization. So for example, we do a Tikhonov regularization or an SVD truncation of our imaging matrix, and that's going to give us an estimation of the sources. And this is often referred to also as like an imaging approach, because the solutions we end up with look something more like this, as opposed to the dipole fitting model, where we identify the best two or the best three dipoles that explain the data. Here we will have actually an amplitude for each one of those dipoles. And then of course we can run statistical analysis comparing conditions, and this will give us islands of activity, and that helps us in our interpretation. This was a very, very brief roundup of source estimation. Of course, it's much more complex and it would take much more time, but basically just to give you a sense of the type of mathematical problems we deal with when we're doing source reconstruction with MEG. Again, connectivity analysis. This is also something that would deserve also a full course just on this, but I'm just going to tell you that there are many metrics that we can use. The electrophysiological signal is very rich because it has a lot of information in the time domain, it has a lot of information across its different frequency components. So technically we use time domain correlation analysis, and we use frequency domain metrics. Some of these are, for example, coherence, phase lag index, and amplitude coupling. We have also a wide range of directional measures, such as range of causality and partial directed coherence that we can use. These are really just some examples of the list of over 10 or 15 techniques that you can apply. Again, because the signal is so rich, so you can tap into different things when you try to explore connectivity. Once you've computed these measures of connectivity, what you can do after that is basically very similar to what you could actually do, for example, with fMRI data. Because this is going to give you a connectivity matrix, be it in the source space or be it in the sensor space. And then, for instance, what you could do is you could identify a threshold, and that's going to give you a binary matrix. And from there, you can compute all sorts of graph metrics. You can look at properties such as rich club or small weldedness and so on and so forth. The interesting thing here is that you can do that in specific frequency bands, and that gives you much more insight potentially into different components of your signal and what they're doing. So in other words, instead of having just a measure of small weldedness that comes from your data, you probably will compute that. You can compute your graph metrics for the alpha band, for the beta band, for the gamma band, for the delta band, and so on and so forth. And this might give you information about the specificity and the functional role of these different components within your signal. Okay, so that was more or less an overview of some of the concepts that are related to MEG and EEG data processing. Now I'm just going to give you a brief overview of some tools and packages, and this is probably going to be relevant also for you here for this BrainHack School. Of course, I'm going to be limiting anything I say to open source software. I think this is the way to go, so we'll focus only on those. So I've got good news and bad news for this. The good news is that there are many open source tools available for EEG MEG data processing, so that's good. And the bad news is that there are many open source tools available for EEG MEG and data processing. And why is it bad news? It's mainly because you have different techniques, some of them competing with one another, some of them doing more or less the same thing, but not exactly the same thing, and there's actually no golden standard. I think in the field of fMRI, there are also a number of techniques that are now available. Some of them have actually, for example, SPM has played an important part and is the go-to tool for many types of analysis. And that has one advantage also, is that that becomes a technique that everybody uses, and results that come out of those techniques and the parameters that are used are readily understandable by many people. In the EEG and the MEG field, that's not really the case, because depending on the tool you might be using, there might be different parameters that go into your different functions, and so it's not that easy to get into that, to interpret the results. And also it's not that easy when you're starting off. If you're a student and you are getting into the field and you look at all these tools and say, okay, which one shall I use? Most of the time you end up using the ones that your advisor or the lab is using, but is that a good choice? That remains to be shown. So I'm going to give you here an overview. As I said, I started by also adding the web links and so on, and I decided that you probably will be able to find those yourself by Googling them. I just use the logos here. So MNE, Python. This is a MEG and EEG analysis and visualization toolbox that I'll come back to in a second. Many of these you're probably already familiar with, Scikit-learn, VisBrain, a number of tools here. FieldTrip is also a toolbox that is quite often used for a MEG and EEG analysis. EEG Lab, of course. Brainstorm is another one, and SPM is another. I couldn't find the logo for SPM. This is the one they have on the website. So if you look at this image, you'll see there's sort of a cleavage between the left-hand side of the slide and the right-hand side. And in the middle, there's something that is an island. So R is another language for itself statistical tool that is very robust and often used for statistical analysis. But you'll see that there's the frontier here is between what is MATLAB-based and what is Python-based. And so I think it's boring to get into this discussion about what is the best, and it's a sort of a useless discussion about a useless battle. But I do have to say that what I think is happening is that actually you had MATLAB walking its way, and all of a sudden Python came, and this thing happened to MATLAB. But don't tell everybody I said that. So Hi, internet. So I'm going to give you a couple of words about MNE. So MNE Python is a software developed. Actually, the original version was developed in Matti Hämäläinen's group many years ago. It was in C, and then when Alexandre Grandfort, among others, joined Matti Hämäläinen's group, they started a Python version of that same MNE code. And since then, we always refer to MNE Python, but I think nobody uses the MNE C, or maybe Matti Hämäläinen himself still uses the C version, but I think everybody switched to the MNE Python version. It's an open source package for exploring visualizing and analyzing human neurophysiological data. So it can be MEG, it can be EEG, and it can be intracranial EEG. It has several modules, and it also nicely links with scikit-learn for machine learning analysis. You can find down here the links for MNE. You'll see that there's an example gallery with many examples for code for input and output. Also all sorts of different examples for pre-processing your data that you can look around into. And when you click on these, you'll also be able to download the Python code and also download the Jupyter notebooks to play around with them directly. As I said, one of the nice aspects of this is it connects nicely with scikit-learn and has an array of machine learning tools available for decoding, encoding, and MVPA. Here we go. So now a few words about some other tools, most of which, or actually all of these tools were developed in our lab. NeuroPython, BrainPipe, VisBrain, Sleep, TensorPack, Spinky. All of these are open source and are available and are in Python. They're available on GitHub. So I'll just give you a quick word about BrainPipe, TensorPack, and VisBrain. So BrainPipe basically has been developed by Etienne Combrisson, a PhD student of mine, and the idea here was really to achieve this interface between neural signal processing of EEG, MEG, and intracranial data on one hand, and the facility to link that to scikit-learn to run some machine learning analysis on your data. So you'll find a range of tools in there that are useful, and it's documented. As I said, it's available on GitHub, and if you have questions, you can contact people from my lab or also Etienne directly. Another tool is TensorPack. The idea here, those of you who are familiar with PAC is phase amplitude coupling. It's a tool that is often used in EEG and MEG, where you look at the relationship between phase and amplitude. So most of the time it's phase of a lower frequency signal like delta or theta, and the amplitude of a higher frequency signals such as gamma. And so there are a few tools out there, but this tool is more versatile, it's faster, it uses tensor computations to compute the PAC, and it has many options in it. Etienne is currently writing up the paper to publish this, but the code is of course already available. If you go for TensorPack, you'll find it on GitHub, and so you can apply it to any sorts of electrophysiological data. For visualization, a tool that Etienne also developed in the lab is VisBrain. It has several modules. It allows you for fast computations using GPU speed, and it also has the ability to use a wide range of data formats. So that's actually something very useful, and it's mainly used for signal visualization on the brain. But it can also be used. There are some tools that we use for example, the analysis of sleep signals, detecting important events in sleep data. So there's actually a module dedicated to that called Sleep, which is here. So if you're working with sleep data, you might want to check out what the module Sleep within VisBrain does. The documentation is there and available. Here are some example figures. You can also handle non-electrophysiological data, such as MRI data, for instance, and I think probably also fMRI with this tool. You can also plot connectivities. You can also plot, this I think was done with MEG and EEG, and you can look at deep structures. There's a wide range of parameters that you can play around with for visualization of the data, so it's actually quite efficient. There's a paper that has been published on this. I forgot to put the link here, but you'll find a full description of this also on the documentation and on the paper published in Frontiers in Neuroinformatics. And this is the module for the sleep data. It allows you also to take annotated sleep, look at the hypnograms, different stages of sleep, and so on. So if some of you are asking, well, some of this looks interesting, and who can help? Here are some of the people in the lab that can help. So Etienne Combrisson and Annelise Sèvres could probably help you with the BrainViz. VisBrain, and for the BrainPipe tool that I mentioned, as well as intracranial EEG analysis with these. For MNE Python, and I recommend that you can either contact Jan, who is somewhere, there he is. So Jan has worked quite a lot with MNE Python, and also Maynak Jas, who was in the lab last year, but now he's with Maternal in Boston, but he's one of the programmers also of this toolbox, and he's generally quite fast to respond, so you can also contact Maynak for this. Last but not least, if some of you are interested and you don't know where to get data from, there are many resources for open data. I did not list any of EEG, you'll find a lot of EEG too. For MEG, this is the Human Connectome project. I think there are about 90 or 95 MEG data sets with there, and also fMRI. Most of the MEG data there is resting state MEG. There's the Omega database, it's resting state MEG, Montreal collaboration with data that comes both from UDM and McGill. The CamCan dataset, this is a dataset of over 650 MEG individuals, resting state but also task. And there's the MNI Open IEEG Atlas for intracranial EEG, and this was released I think last year. And there are many more. I don't know how well I'm in time. I hope I have a few minutes if you have any questions. So that was everything I wanted to tell you. So have a good time hacking. I hope you enjoy this Brain School.
So we are doing fantastic with time.
Good.
Congratulations. We still have 10 minutes before we have to start yoga in 10 minutes, so maybe we can take like 5 minutes of questions. If there are any. Yes, no?
Yeah, so I work on in the G lab for sure.
Yeah.
So I want to convert in MNE. I don't know which toolbox exactly to work on phase lag index.
Yep.
And machine learning, so like two cluster, let's say two groups. So what is your advice of the tool I should use? Is it in MNE, or should I go to brainstorm, or should I go and...
Okay, well, so it's a tricky question. I would say, depending if you know that you're going to afterwards take your data and you're going to continue doing classification using machine learning algorithms, yes. I think for machine learning, there's really not much doubt in my mind that Python-based tools are the way to go, and toolboxes anyway. So you're going to find yourself using scikit-learn anyway, okay. And getting familiar with Python anyway for that. So in that case, the step before, if you're hesitating between a MATLAB-based tool such as Brainstorm or Fieldtrip, or a Python-based toolbox like MNE, if you know you're going to be using Python at the end of the day with the machine learning, then you probably should anyway go with MNE. And also MNE has a nice, an easy way to interface with scikit-learn. So there are several tools within MNE already that allow you to do classification and modeling.
And it's BrainPipe.
BrainPipe is a separate tool that's also in Python that's developed in our lab.
Okay, but
but yeah, exactly. But it doesn't do, for example, what MNE does is, for example, source reconstruction for if you have MEG data.
Okay.
If you have EEG data and you already have your signals and that's what you want to classify, you want to compute PL, for example, phase lag index on that, then you could probably also, BrainPipe would also be definitely something you can do, and you'll have people around to help you.
Thank you.
Yep.
So you were presenting two methods for source reconstruction. So there was a dipole fit and distributed sources. Is there a difference in the way we use these two methods? For example, for one experiment, we are really interested into what happens in these seeds regions, so we will maybe favor one method over another, or what is the practical differences between the two?
So I would actually never go with the dipole fit unless what you have is a very strong, for example, primary visual or primary motor response for which you have a strong ERP. That's really nicely identified in time, and you have a strong, and what you want is the component that is responsible for that. But as soon as you're interested in like more cognitive, complex tasks, if you're looking for a task like in your case into attention and things like that, it's more distributed. You're also interested in looking at connectivity between brain areas, and in all these cases I would favor imaging solutions like distributed sources. So minimum norm estimate is one approach that I think is quite good for that.
All right. So, any other pressing question for Karim? Otherwise, thanks, Karim,
Okay.
again.
Thank you very much.
And as per usual, like this is the advertisement for the science, but not the science itself. When you actually start, dragging some data and processing some data, if questions arise, you can contact Karim on
Yep.
Slack, Jan on site, or other people you listed from your group. And also there's a Mattermost BrainHack. You have had this link. There's quite a large community on there. You can also ask for help. Oftentimes there are folks online who can through direct messages interact with you and sort things out if you cannot find a resource directly in our local sort of mini BrainHack Slack, and before you jump to other forums. It's always a possibility. So yeah, we've got a 10 minutes break, and we should have yoga, I believe, on the terrace because the weather is good. So I'm going to go help set up, and I'll see you there. So.
J'étais dans mon temps là. Je suis encore sur devant mon ordinateur. Ok, vous êtes où?